Hi, I'm Louise Stevenson. I'm a third-year PhD student in the Ecology, Evolution, and Marine Biology department at UCSB. My advisor is Roger Nisbet, and I'm from Dallas, Texas. We're specifically looking at freshwater green algae, so a really common model organism, and then Daphnia, which are these little filter-feeding crustaceans that actually eat algae. So we're looking at the effect of these engineered nanoparticles on each species individually, but then also looking at the effect when the species interact. So trying to look at a more environmentally relevant system, because in the environment the species don't exist on their own. Our model system is a basic freshwater model ecosystem like you would see in any lake or stream. So we have bacteria in our mesocosms, although we aren't studying those as much as other groups. Bacteria compete with our single-celled algae by taking up nutrients, so the algae are growing by taking up nutrients that the bacteria are also using to grow. And then we also have Daphnia, which are filter-feeding rotifers that actually eat the algae. So we have a tritrophic system, or 3 food levels, where the Daphnia are eating the algae are competing with the bacteria to take up nutrients. And so all of those interactions can be affected by nanomaterials. So we're looking at the effect of how the nanomaterials can directly affect each species, and then how they affect all of those processes in conjunction. Okay, so one of the most exciting findings that our group has come out with is that we actually looked at the effect of silver nanoparticles on an algal species at different stages of growth. And what we found is that at later stages of growth, so after the algae have spent 2 weeks dividing and metabolizing, so basically using nutrients to make their populations bigger and to grow and photosynthesize, all those processes, the byproduct is a dissolved organic carbon, which is basically just a big organic molecule, and those big organic molecules actually make the silver nanoparticles less toxic. So that as the algal populations are getting larger, they're actually protecting themselves from the silver nanoparticle toxicity. We're interested in looking at the effect of nanomaterials over ecologically relevant timescales, but also over the entire population and even on interacting populations. So a species is not going to interact with nanomaterials on their own. They're going to have other species around that they're interacting with. And so that's the big important finding behind our most recent publication, which is that if you looked at the effect of, say, silver nanoparticles on Daphnia just in regular media, so doing a standard toxicity test, throw in some Daphnia, throw in some nanoparticles, see when they die, you're going to get a completely different answer than if you actually consider, if algae were present as well, producing organic compounds which can have this protective effect. I specifically knew that I wanted to do ecotoxicology, which is kind of the broader field that we're working in. In college, I went to Amherst College in Massachusetts, and it was a very small biology department. The school is very small. It's a great school, but it had about a dozen biology faculty members. And I ended up working with a professor who was looking at the effect of a different toxicant on fish. And I didn't think it was something I would be interested in, but I really liked the professor, and so I signed on for this project. And it ended up being really cool. Like, I really liked the idea that you can take a fish, you can see the effects of these environmental pollutants and how something in the lab can actually relate to something that's happening in the real world. And that kind of, you have the advantages of small-scale manipulation, being able to control everything in a lab, but it could actually have environmental implications for the world outside. If our lab caught on fire, I guess the first thing I would rescue would be, we have a data server that has all of our data on it, and I would rescue that first, which is a really lame and nerdy response, but that's actually probably the first thing I should rescue, once I made sure all the people were out.